It is now my pleasure to introduce the speaker, Dr. Leonardo Ferreira. Dr. Ferreira is an assistant professor of microbiology and immunology, and by courtesy of regenerative medicine and cell biology at the Medical University of South Carolina and the Hollings Cancer Center. He holds a BSc in biochemistry from the University of Coimbra, Portugal, a PhD in biochemistry from Harvard University, and completed a postdoctoral fellowship at the University of California, San Francisco. He is fascinated by the phenomena of immune tolerance, and his long-term goal is to control how the immune system defines self and non-self. Such knowledge will allow the design and development of powerful new engineered immune cell therapies to fight autoimmune disease, cancer, and aging. With that, I hand it over to you, and you're welcome to start your presentation when you're ready.
Thank you. Thanks for the invitation to speak today. I'll start actually by saying that Vector Build has been a great partner in starting my own laboratory since 2021 to design all sorts of constructs, as you'll see some of them here. So it's really a treat to be part of this. So today we'll be talking about using our own cells as living therapies for all sorts of diseases. And so this idea started a long time ago. Centuries, millennia ago. And so the simplest way, or the most widespread way that this comes across, is in organ transplantation, right? In organ transplantation, which has been a technique tried for a long time, but it's really all in the 1950s that it really came to fruition. This is 1954 at Harvard Medical School, the first successful solid organ transplant. It was done between identical twins, was a kidney transplant. And so we've come a long way since that time, but the basics are still there. So basically replace damaged cells, tissues, organs with new ones. Now, this need is in many cases, and a very big one is autoimmune disease, where your immune system is actually attacking your own tissues. And for instance, in type 1 diabetes, the immune system of these patients is recognizing and destroying their insulin-producing beta cells. And so you see here a time lapse over time. As the disease progresses, your insulin-producing beta cells, marked in red, they disappear over time due to infiltration of immune cells, which are marked in this gold color. And so you see towards the end, there's almost no beta cells left. And that leads this type 1 diabetes patient to totally dependent on exogenous insulin injections. And as you know, these are necessary to control blood glucose. If you don't watch for your blood glucose levels, you can go blind, you can lose a limb, you can even die. And so it's a serious disease, affects millions of people, starts as children, adolescents. And so it would be great to understand the immunology of it, and also how can we take this idea of using cells as living therapeutics to help with this disorder. So we know that it's caused by the immune system, and the key players in this attack are T cells, T lymphocytes. And so what we know is that type 1 diabetic patients have these autoreactive, these self-reactive T cells that recognize and destroy the pancreatic beta cells. They set on fire, if you will. And so that will then prevent it from working. Now, there's a solution to this, which is, well, if your beta cells are gone, just put more in. And so one way is to do pancreas transplant. More recently, what people realize is that, well, in a human pancreas, only about 2% of it is the endocrine system, the islets of Langerhans. And so instead of taking a whole pancreas, the current iteration is to first digest the pancreas, what's called a Ricordi chamber, and take the islets from it. So the islets will have alpha cells, beta cells, and delta cells, and then infuse them through the portal vein into the recipient patient. And so what you will see here is that if you do positron emission tomography, PET, for glucose consumption, you'll see nicely in this patient, an example, this is from University of Pennsylvania, that the islets are engrafted onto the liver of the patient. And so luckily for us, beta cells, as long as they have a steady blood supply, they can compute blood glucose and secrete insulin accordingly. So we know it's possible to replace the lost beta cells. Now, of course, there's an issue, is that this all started because it was autoimmune attack. So just putting more beta cells in on their own will be like building a sandcastle by the ocean. The waves will come and wash and destroy it. So then this requires a lot of immunosuppressive drugs. These are blanket drugs. You might have heard about cyclosporin, tacrolimus, steroids. and they just shut down the immune system as a whole. And so while this indeed prolong the life of this foreign graft, right, who's taken from someone else, from a deceased donor in this example, they have a lot of side effects. They're toxic to the kidneys, they increase your blood pressure, toxic to the liver, and especially problematic in children. So remember type 1 diabetes being diagnosed in children, adolescents, because the longer you live taking these immunosuppressive drugs daily, the more likely you are to suffer toxic side effects, neurotoxicity, for example, so learning impairment and also growth impairment because of the steroids, for example. And so it would be great to have beta cells going in without needing these immunosuppressive drugs. That will be the ideal, the goal here, the holy grail of immune tolerance, to have accept this organ as their own. So how can we do this? Well, luckily for us, not the entirety of the immune system is evil in this case, right? As you know, the immune system will, fight pathogens, fight cancer cells. In autoimmune disease, also fight some of your own tissues, but so can use some of it to actually protect. And so within these T lymphocytes, they're not all the same. There's actually a subclass, a subset of T cells that their job is not to kill other cells, but actually prevent killing from happening. So this was found, hypothesized in the 70s, the 80s, was really in the 1990s that we got a molecular handle on this. And so there's these key experiments in mice first, where if you have a mouse and you thymectomize the mouse, remove the thymus away, that's where T cells go to school. And if you do it very shortly after birth, these mice have all sorts of autoimmune disorders, gastritis, ophoritis, thyroiditis, type 1 diabetes. And so there must be something developing in that very first stages of life that is essential to maintain tolerance to self, right? Our immune system does not kill our own tissues. And so those turned out to be these regulatory T cells. They are characterized by expression of this molecular marker on their surface called CD25. So CD25 is the IL-2 receptor alpha chain, IL-2 being a growth factor for T cells. So at the time, people were wondering, how come that these cells that are required for us to not destroy our own bodies, why do they have CD25 and what are these cells? And so a lot of decades have gone by, and so now we know a lot more about these cells. We know developing a thymus, so it makes sense why then removing a thymus would cause their disappearance. And this has a lot to do with how you select for T cells. So the thymus, as I mentioned, where T cells go to school, what I mean by that is that every T cell is a unique T cell receptor that allows you to see the world in a unique way. And so not all T cell receptors are equal. If they bind to, the binding is too low, not strong enough to target, the cells die, death by neglect, then you're good. If they bind too tightly to yourself, that's dangerous, right? The self-affinity is too high, and they're negatively selected. The regulatory T cell sits somewhere in between the middle ground and a bit high, where these cells, instead of being killed, because again, they could be autoreactive T cells in the future, they instead turn on expression of this transcription factor, so protein that binds DNA and makes decisions, called FOXP3. And with FOXP3 expression, that comes C25, which I just mentioned, the first marker to be discovered in relation to Tregs, but also other markers such as CTLA4 and others. And so this is where the cells come from now. Very broadly speaking, the way they suppress, inhibit immune responses, there's two main tenets. One is bystander suppression, meaning that when the regulatory T cells in a specific environment, no immune response is able to take off if the cells are there. And the other tenet is infectious tolerance, meaning that when a Treg goes in, it not only inhibits immune responses at the local microenvironment, it also convinces other naive, undecided T cells to themselves become, in the future, regulatory T cells. And so these are two main ways in which Tregs work to build immune tolerance to self. Now, at the molecular level, there are, I think, now 16 different mechanisms that Tregs accomplish this. I'm just touching 4 very broad categories here. One is cytokine secretion. So cytokines are soluble mediators, and Tregs will secrete anti-inflammatory cytokines. So they will shut down specific immune responses. Also C25, which I've mentioned several times now, the way it works is it sponges up any extra interleukin-2 IL-2 that is around, that does not allow T cells from ever mounting an immune response. Tregs can also reprogram, if you will, the immune microenvironment. For example, exogenous ATP is a danger inflammatory signal. Why? Because ATP belongs inside the cells. So our body has evolved ways to recognize ATP as a danger signal. So Tregs have these ectoenzymes on their surface to actually convert ATP into adenosine, which then basically puts T cells to sleep. And then last but not least, Tregs sometimes take matters in their own hands, and they can actually directly kill antigen-presenting cells. So one way to shut down an immune response is by eliminating the cells presenting the peptide to the aggressive T cells. And so in all these different ways, Tregs either modulate antigen-presenting cells, or they modulate the T cells or the B cells. They're all different ways make Tregs kind of hard to match. It'd be very hard to make a single small molecule or a single biological that would mimic this polypharmaceutical capabilities of the cells. And so that led some of us to believe that then maybe we can use Tregs themselves as a living therapeutics to induce immune tolerance. So still the scales from situations where there's too much immunity, too much immune attack, such as in type 1 diabetes or an organ transplant in general, to an environment of regulation. Everything is under control. These beta cells belong to us, they're being accepted, and other organs for other diseases. So how far are we in fulfilling this vision? So there's several trials in humans and then also preclinical studies. So in humans, we're not too far yet. So we know that these regulatory T cells, it is advantageous to partially substitute these blanket immunosuppressive drugs, such as tacrolimus, steroids with Tregs. You see here the main advantage being that you have less infectious diseases in the patient. So you see less pathogen in the regulatory cell group compared to the control group, and also less virus infections, but it's not sufficient to completely replace the immunosuppressive drugs. Part of the reason why is because these Tregs, they are rare in the blood, number one. Number two, if you take Tregs from people, from transplant patients in this example here, that have already been undergoing immunosuppressive drug treatment, their Tregs are already weaker, for lack of better term. So they'll grow less, and when you infuse them into patients, inject them intravenously, they also seem to go down over time. And so some challenges in the manufacture and survival of these Tregs, just taken straight from blood. So then how can we go about this? How can we improve on this? Well, so one way is, can we genetically modify these Tregs to be more laser focused, if you will, right? Right now, in these examples, this cells is just a mixed bag of specificity. You take someone's blood, isolate all the regulatory T cells, expand them in a laboratory, put them back in. And so there's no knowing really, or it's hard to calculate how many of these Tregs are actually active, how many of them go to the transplant of organ and protect that organ from immune rejection. So we need to somehow overcome this, but these cells are rare, as I mentioned to you. So then one potential solution is, well, why don't we make our own specificity for these Tregs? And so one way to do that is to learn the biology of the T cell. And so you see a T cell usually gets activated with signal 1, which is T cell receptor mediated. And then signal 2 is C28 co-stimulation, or encouragement, as I like to call it. And so T cells need both these signals to get activated, spring into action. So we can mimic this with what's called a chimeric antigen receptor, CAR for short. And so you might have heard of CAR T cell therapy for cancer, right? Where you're redirecting T cells to attack specific surface proteins that are expressed only in cancer cells. So this receptor then allows you to repurpose, to redirect a T cell to any target you want, as long as on the surface of a cell. And so the way it does is you have an antibody-derived component. So the antibody part is what sees the target you care about. And then the intracellular domain, what's inside the T cell, is mimicking these natural signals. So you see the signaling domains here, these peptide sequences provide the T cell signal 1, signal 2 upon encountering this antigen of interest. So taking that CAR, can we then take Treg therapy to the next level, make it more efficacious, more targeted, with better benefits? So what should we target though? Right, so we have the cell type is the regulatory T cell to induce immune tolerance. We have a targeting mechanism, which is the CAR, but now what should we target it to? What would be a good antigen? So if you look at transplantation, a big barrier to transplantation success is the genetic diversity between people, specifically in terms of their HLA allele expression, so human leukocyte antigen. This is really what determines self, non-self recognition, success and failure in transplant. And so we wondered, so usually it's bad news when there's a mismatch in HLA, but we wondered, can we turn the table around and can actually have a mismatched HLA be not a bad thing, but actually a good thing? So an HLA is a unique marker for the transplanted organ. So for example, if you're a type 1 diabetic patient and you receive islets from a donor that is HLA-A2 positive, and you yourself are HLA-A2 negative, now HLA-A2 becomes a unique marker for the transplanted islet. So it stands to reason that if I target Tregs using CAR, an HLA-A2 CAR, to this target, I can then make Tregs that specifically go to the pancreas and protect it from rejection by the recipient's immune system. So exactly what we did. We made HLA-A2 CAR Tregs that would then inhibit these autoreactive T cells against the pancreas. And then ideally that would then prevent inflammation from taking place. And so now we restore the capacity of these patients to make insulin, then therefore controlling their blood glucose without requiring any exogenous insulin. So this work, I'll have a couple of vignettes. This work first started at the UCSF when I was a postdoctoral fellow in the laboratories of QZ Tong and Jeff Bluestone, and had a very close collaborator who himself is also faculty now, Yannick Mueller in Switzerland. And so what we did though was we took the Tregs from human peripheral blood and we used CRISPR-Cas9 to insert in their T cell receptor gene locus a chimeric antigen receptor, a CAR, specific for HLA-A2. And so this is the workflow here. We get blood, get the cells, stimulate them, and then do the CRISPR-Cas9 engineering. And so what we saw is that you see here successful insertion of the gene in the genome. So CTCR-C3 complex goes down if you do the knock-in successfully in that locus. And then the MIC tag is a marker for surface expression, cell surface expression of the CAR. And so you see nicely that at roughly 2 in 3 cells became edited in this way. So that's step 1. Step 2 is, of course, confirm that they are HLA-A2 negative. Of course, the Tregs will just, bind and activate themselves. Another important step is to make sure that these cells are still Tregs. And so, as I mentioned to you earlier, FOXP3 is a master transcription factor and marker of Treg identity, and Helios is another important transcription factor. And so the gold standard in the field is that a Treg is FOXP3 Helios double positive. And so you see nicely here, if you take conventional effector T cells, these are the inflammatory cells, they're double negative for the most part with some cells that are FOXP3. But then if you have the Tregs either edited or not edited, they are 90% and above FOXP3 Helios double positive. Okay, so we have the CAR on the surface of the Treg, and it's still a stable Treg. Now, does it get stimulated? Does it work? And so for that, we put in some B cells that have HLA-A2, and you see nicely upregulation of the activation marker C71 on the Tregs. So we have Tregs that are on the surface, they remain Treg identity, and they are activated in the presence of HLA-A2. Now, in this business, we want to know not just what they're doing in vitro, but also where do they go, right? So they have to go to the islets. And so what we did there, we took NSG mice, these are immunodeficient mice, and we transplanted human islets, HLA-A2 positive islets, in the kidney capsule. So we put the mouse on the side, and then we transplant the human islets right there under the kidney capsule. So highly vascularized site. And the mouse, also easy to find, localized. After that, it's having grafted, we then do an immune challenge where we inject either polyclonal Tregs or HLA-A2 CAR Tregs. And they have this luciferase gene. So basically the cells glow in the dark if you inject a substrate to the mice, so we can image where these CAR Tregs are inside the mice without hurting the mice over time. And so this is what we saw, very black and white. If you just infuse polyclonal Tregs, so mixed bag of specificities into these mice, you see at the beginning, so it was a tail vein injection. Yes, at the beginning, they go tail vein, they go to the spleen, right? On the left side of the mouse is a spleen. But then very quickly, less than a week later, you don't find them anymore. And so this is probably what happens also in patients. They receive polyclonal Tregs that did not meet efficacy, is that there's too many specificities. Very few Tregs really go to the right place. But if you genetically modify the Tregs with the HLA-A2 CAR, now you very nicely see they go intravenously, they go to the spleen first, where immune cells like to hang out, and then over time, they migrate, they traffic from the spleen on the left side of the mouse to the right kidney capsule where the transplant happened. And so you see there, even past 2 weeks later, 5 out of 6 mice still have this very nice localized signal of the CAR Tregs in the islets. And so this shows that this is living drugs that really go where they're needed. Also, importantly, they're safe, right? The trafficking of these CAR Tregs to the islets does not compromise their function. These mice remain normal glycemic after the injection. And as a nice control, if you put the same CAR receptor in conventional inflammatory cytotoxic T cells, then you see that most of the mice, immediately the islets get destroyed, and the blood glucose goes through the roof. And so in the conventional control here in the black lines, you see, vroom, like in 5-7 days, islets are destroyed. So again, it's a very potent CAR. But if it goes in the regulatory T cells, now there's no safety issue. Okay, so we have that. We have a receptor, and they go to the right place. Now, do they work or not? That was the question, right? And so do they protect from immune rejection? Also, what is the best target, generally speaking? You know, HLA-A2 is going to work for some people. Roughly a third of the population has HLA-A2, but some people don't have HLA-A2 and or they have HLA-A2, and therefore they cannot receive transplant as HLA-A2 and use this technique, because then the CAR Tregs will just go everywhere in their body. So can we make a more universal target for these cells once we show that this really works? And so this is where I got really fortunate to have a collaboration. Dr. Holger Russ at the University of Florida, he's still our postdoc, Jesse Barra, and in my own laboratory, Rob Rubino was a technician who's now in a PhD at UPenn. And so what we did then, we really went back to the whiteboard and thought, okay, we have the Tregs, we have the CAR targeting technology. Can we show efficacy in these humanized mouse models? And can we get to decide, you know, not just the CAR, but also the target, not just the receptor, but also the ligand? And so how can we go about this? Well, step one is the source of beta cells. So instead of limiting ourselves to beta cells from cadaveric donors, can we actually just make them in a laboratory? And so this is where the Russ Lab was really instrumental, because they make human pluripotent stem cells become beta cells. And so with this, you can then make ideally unlimited supply. So beta cells is no longer a problem to have enough of them. And so I didn't mention, but currently you have to pull together islets from 3 to 4 donors to go into one recipient. So if there's already, imagine, so if there's a kidney shortage for organ transplant, where each donor can give kidneys to 2 people, right? So it's 1 to 2. Imagine how bad it is if the ratio is instead of 4 to 1. You need 4 people's organ cells to go into one recipient. So very key to be able to just make beta cells in the lab. Now, of course, this is true, and they respond to insulin in glucose in vitro, but they still have this challenge of, if you just put them in without any immune protection, they're going to get killed. That's where the Tregs come in, specifically the CAR Tregs. There's this promise that it can actually inhibit multiple cell types that could try to attack these precious beta cells. But of course, there's no without challenges, one of them being, what's the target, right? Where is the CAR Treg recognized and is unique to the beta cell and nowhere else, and can work for everybody? And so we thought, if we're starting from stem cells anyway, can we genetically modify them to make our own ligand, our own target? And so if we could, what should this target be? It should be on the cell surface. It should be biologically inert. We don't want to hurt in any way or form or shape the function of the beta cells. It should also be immunologically inert, right? So now our body is very keen on finding foreign molecules. So we can't put any like molecule that comes from a bacterium or a virus that will cause rejection problems. And of course, perhaps most importantly, be unique. It should not be found in any other cell types. So for our first then proof of concept, what we came up with was a truncated form of EGFR. So epidermal growth factor receptor. And it's truncated, meaning that there's no biological function, no signaling domain. And it also opens up this unique, by truncating also part of the extracellular domain, there's this unique domain region that can be recognized by an antibody. And so that's our ligand to start, is this truncated EGFR that will go on the stem cells and then consequently on the beta cells. And on the Treg side, we have an EGFR specific CAR. So what we're doing is we're making these CAR Tregs recognize, talk to the stem cells expressing this ligand. So can we show this proof of concept? And so on the stem cell side, we did the CRISPR editing just like in the Tregs before, but now in the AVS1C locus. And so we put in the truncated EGFR and also luciferase gene, because as I mentioned earlier, that's helpful to track the cells inside the NSG mice. And so we do that, again, collaboration with the Russ lab. And so we see that these stem cells are still pluripotent. You have FOXP3 and Helios as identity markers for Tregs. In the stem cell world, the counterparts, if you will be OCT4 and NANOG. And of course, EGFR T expression is super high in the edited cells, but not in the wild type. So we show that also by flow cytometry. And then they also have luciferase, right? So if we put them into a humanized mouse, we will be able to track the graft mass without having to be invasive in the mice. So that's the stem cells. They differentiate into beta cells unimpeded. So that was, kind of the checkbox number 2. And the answer is yes, it is okay to express this ligand on the stem cells. They make beta cells same exact efficiency, and the beta cells have just as much C-peptide, and there's just as much NKX6.1 transcription factor expression. So we have EGFRT expressing stem cell-derived beta cells. Now, and they also function, right? So if you give them glucose, their insulin spikes up, indistinguishable from wild type. Now, on the Treg side, take them from blood, transform them or transduce them with this CAR, and then they remain Tregs, again, very important. So we have stem cells and beta cells with a ligand, and we have Tregs with a CAR receptor, and they get activated by the ligand, right? So they talk to each other. That was checkbox number 3. And the answer is yes. You see C71 upregulation, also C69 another activation marker. So you see only when the CAR Tregs are together with the stem cell-derived beta cells with EGFRT do you see upregulation of this marker. Versus alone, you see it's very low. So they're talking to each other, and they're making anti-inflammatory cytokines. I mentioned IL-10 before. There's also others as well. So again, we have beta cells and Tregs, and they have their pieces. Now, we have a couple data to make sure it's really safe. So no matter how many times we stimulate these Tregs, they remain Tregs. FOXP3 Helios double expression very high, and they're not cytotoxic. So this is just in vitro first to see there's no killing by CAR Tregs. So they're safe, and they talk to the ligand. Now, they also don't secrete inflammatory cytokines. There's no potential toxicities. So now to the real experiment, which is, do they work? So first, in vitro, we show suppression. So if you co-incubate these CAR Tregs with effector T cells, you see nicely that in the presence of the stimulated EGFR CAR Tregs, T cells, both CD4s and CD8s, divide much less. So you see these are Treg to effector ratio, so it's a dose response curve, right? The more CAR Tregs you add to the co-culture, the more suppression there is, because the less the T effector cells were able to proliferate. Tregs work, as I mentioned earlier, not just directly on T cells, but also on antigen-presenting cells. And so I also made sure to look that the CAR Tregs are downregulating co-stimulatory molecules on the dendritic cells. And the answer is they are. You see here CD80, CD86, which is bound by CTLA4 on the Treg surface, and you see it gets ripped out. You see downregulation there. So these CAR Tregs are inhibiting both T cells and antigen-presenting cells in vitro. Now, how about in vivo, right? Are they protective or not? And so for that, we do as before. Now, instead of being cadaveric islets, these stem cell-derived beta cells, but also in a kidney capsule, wait for that to engraft, and then do the immune challenge. And so here we're doing 2 conditions to have the proof of concept. You either give in the killer T cells that recognize the beta cells, and so these cells should reject the beta cells, or we're combining them with CAR Tregs. And the question is, do the CAR Tregs prevent the rejection of the beta cells, which is what we want to accomplish in patients. And so what you see here very nicely is that if you give the CAR T effector cells alone, by day 2 weeks, gets lower, almost no sign left of the beta cells in these mice. But if you co-treat with a CAR Tregs, now you see that the graft mass is maintained. So this is quantified here over time. You see, you go out to like almost a month later, and you see the beta cell mass is preserved in the presence of the CAR Tregs. Now, of course, seeing is believing. So first are the CAR Tregs in the sites, because that's part of the premise is that the CAR Tregs go where they're needed. And the answer is yes. If you take the graft out and do flow cytometry, you find very nicely the FOXP3 expressing Tregs specifically in the mice treated with CAR Tregs. And of course, what does the graft itself look like? And so this is probably the most beautiful image that we ever saw up until this point, was that you see, if you put the T effector killer cells alone, very few and far in between insulin cells left, a lot of infiltrating CD3 positive T cells, very few to no natural occurring FOXP3 Tregs. But if you treat with a CAR Tregs, now you see beautiful insulin expressing clusters of stem cell-derived beta cells. You still see some T cells, yes, but most of them are FOXP3 Tregs, which is exactly what we wanted. And so we see that if you look for EGFR T, also it's maintained. So the target is not lost in the transplant, which is important because you want the symbiosis, if you will, to be maintained over time to induce immune tolerance. And then this will be an overlap to show you that insulin expressing beta cells are also the ones that are expressing the EGFR-T unique ligand for the CAR Tregs. And as HNA is a human nuclear antigen, so it can pinpoint the human cells in the mouse tissue background. So here's a quantification of what I just showed you. So again, almost no Tregs in the control mice. Half of the T cells in the treated mice are Tregs. You see a way more EGFR-T expressing cells as part of the human cells in the mouse graft. And you see also reduction in T cell infiltration, more than 50, more than 100%, right? Less than half of Y2B. And so we show that the CAR Tregs, they infiltrate, they suppress T cells, and they preserve the engineered insulin-producing beta cells. So we basically made the lock and the key to induce localized immune tolerance. So now in the next 2-3 minutes, I'll just go over, this is what we have and shown. We've published this work very, very recently. Now, what's going to happen next, right? What's the vision for the immediate and late future? So one thing is, so the current version of this would be a mix of an off-the-shelf and personalized treatment, right? To have off-the-shelf engineered stem cell-derived beta cells with a unique ligand, and then you take the patient's blood, take Tregs out, modify them with a CAR, and make them recognize, become the bodyguards of those stem cell-derived beta cells. Now, hope I showed you this. We don't have to go through all these texts. Now, what's next, right? So one thing is to have even a more rigorous humanized mouse model. And so there's a lot of work. I'll either reference it just for you to see in your own time, but so there's many ways in which we can model human type 1 diabetes in mice. And so some ways are to use the latest version of a human gene knock-in mice. So you can knock in these 4 human genes to basically improve the engraftment of human immune cells in the mice. So now you can have more myeloid cells and natural killer cells and others. That's one aspect. The other aspect is to use T cell receptors from type 1 diabetes patients and see how their rejection of the islets compares with just our CAR control. And then, of course, point number 3 is we want to mimic the inflammation, high glucose environment of diabetes as much as possible. So you can induce diabetes in mice using streptozotocin, which will kill their endogenous beta cells, and it can give way for these human beta cells to really have to put in the work, right, to rescue the diabetes in the mice. And then how does that interact with the protective CAR Tregs? So those are some, technical next steps. The other thing is the target. So EGFR truncated version, it was a great proof of concept, but it could have some cross-reactivity. So your skin, as you might imagine, has a lot of EGFR. So can we have our different target instead? So we've developed our own translationally relevant unique CAR antigen that obeys all these properties. So what we've shown so far is that we made some target cell lines to show it's on the surface, which is an important point. And we've also made CARs that bind to that ligand. So again, we made a new ligand receptor lock-in key pair. And so you see nicely here that this CAR is talking to the ligand. And so now a lot of the work we're doing is to basically being able to reproduce that. We can see with this unique translation relevant ligand CAR pair, the same results we saw with the EGFR truncated and EGFR CAR. Another aspect is the design of the CAR itself. So I showed you an example where we have EGFR CAR Tregs, no inflammatory cytokines, they suppress T cells, suppress dendritic cells, they protect the islets in vivo, the stem cells or beta cells. Now, not every CAR is the same, is equal. And so what we're finding in the laboratory, in the parallel project, is that depending on the affinity, for example, how tight the CAR binds, the Tregs might behave differently. So we have this recent story that came out of the lab where you see if you have a high affinity versus low affinity CAR, now the high affinity CAR can force, if you will, human Tregs to misbehave. You know, they don't like this tight binding, so they start making interferon gamma, TNF alpha. These are all inflammatory cytokines. And so what we're currently doing is how can we really optimize CAR design to be uniquely tailored to immunosuppressive, long-lived Tregs. And so using artificial intelligence with a collaboration with Dr. Chatterjee at Duke to really design with really molecular precision how tight and where the CAR binds in this new unique ligand that we've developed. And so the other thing is, can we synergize this with work at others in the field? So in the past, I myself have been involved in this idea of making universal stem cells. Like have a universal blood donor, could you make a universal stem cell? And so that will require a lot of gene edits on the stem cells themselves, right? What I showed you is the stem cells are untouched other than this unique ligand, but you could imagine going at it and just, ablating HLA expression so T cells can see it, expressing some NK cell inhibitory ligand. So now the natural killer cells also can kill them. And so there's quite some work on this to make these universal cells. And so again, coming from stem cells, what people have shown in other groups is that you can make these hypoimmune beta cells, and then they've shown both in humanized mice and also actually in non-human primates, and not just in those animal models, but actually in people. So what this is Center Biotechnology is work between Boston, South San Francisco, and Seattle is that there's a patient in Sweden that received these hypoimmune stem cells. So they have no MHC class 1, no MHC class 2, and express C47, which appears to inhibit both the natural killer cells and also macrophages. And what you see is that at least up to a month, these islets are still there. These genetically modified islets are still there. And the patient has some C-peptide. Now, this is not a curative dose, so the patient still requires exogenous insulin injection, but you see the C-peptide level goes from 0 to something. And so it's very exciting technology to engineer also the stem cells. And so then the way I see it then is that can we synergize this, right? And even learning from xenotransplantation, where now you need even more gene edits to make it more compatible with donors. So we can learn from this such that not just the Tregs are being modified, but the stem cells can also be modified to give rise to what we call hypoimmune cells. Now, one last slide is that the Tregs themselves can come from stem cells. So there's no protocols that it can differentiate Tregs from stem cells. And I could imagine, instead of being what I showed you before, the whole therapy could be off the shelf. You have off-the-shelf stem cell-derived beta cells, and you have off-the-shelf CAR Tregs. And so that will really help democratize this strategy. So then with that, there's some outstanding questions, right? I mean, one is the biology that the CAR Tregs face when they're in the inflamed pancreas. What is the best source of these cells? Are we differentiating them from stem cells? Are we taking them from the patient? Are we taking them from a healthy donor, right? I mentioned to you that especially transplant patients, their Tregs are a bit beat up after all the drugs they've already taken. And then, of course, the second part is specific to this project is how generalizable is this CAR ligand pair engineering. So as I mentioned, I have this proprietary CAR ligand pair we're testing on now. And then, of course, can we synergize with our gene edits? Like in my view, I don't think we should completely eliminate immune recognition of stem cells, because if there's a virus infection or cancer formation, good luck getting rid of that graft. And so that would be dangerous. But we can definitely have additional ligands on the stem cell-derived tissues that can either further help recruit and foster the Tregs or also act on other arms of the immune system. And so there's a lot of potential in this field for early genetic modification of both the grafts and the immune system to prevent rejection of the graft. And so with that, I thank you for your time. I have a lot of things to give. I've showed you some data still as a postdoctoral fellow at UCSF in the Tongue and Bluestone labs, also my own laboratory here at the Medical University of South Carolina. Rob and Russell with their name bold because they're the main ones working on the stem cell-derived beta cell and CAR Treg biology. And then also I mentioned collaborations with Duke with a new, potential avenue to use artificial intelligence to design new constructs, which actually they all say vector builder has been key because we've designed, I think, 25 different constructs to really, narrow down what's the motif that might be best to design new cars for Tregs. And with that, I'll be happy to take any questions. Thank you again for your time and attention.
Thank you so much. That was fascinating. We do have a couple of questions. The first one is it possible to prepare Treg from iPSC as well?
Yeah, so that's an excellent point. So this work just came out of a lab in Japan, I think 6 months ago, maybe. And so yeah, almost a year ago now. Time flies. But so yeah, I think it's very exciting, and actually I like it because they really took our playbook of putting HLA to CAR. I didn't show the data today, but we also shows graft-versus disease protection. And so in their work, at least, they basically really compared iPSC-derived Tregs and natural Tregs, and they performed equally well in these preclinical models. So that's a good question. I think there's a good possibility there to be able to generate the Tregs from stem cells, so then you don't no longer need to be sourcing them from the patients. Yes.
Fantastic. One of the issues with studies like this is the suitability of rodent animal models. What alternative approaches are being used to supplement the supportive evidence?
Yeah, that's a great question. So one big challenge with rodents is that, well, one is that their size, of course, but also their lifespan, right? The experiments I showed you, they go over the course of at most a month. But in humans, you want to talk about years, not about months. And so there's a few approaches there. One I showed you again, work from another group to use the non-human primates, right? So this study here where you can do this in primates. This will be a bit harder for our specific case because there's a specific protocol that you use to differentiate beta cells from stem cells. And if you work with macaque stem cells, they behave differently from human stem cells, which behave different from mouse stem cells. And so that's one potential challenge in our specific case, which is, so in their work, what they did was they took the macaque islets and they edited the primary islets. So these are P means primaries. They didn't do stem cell approach, they did primary ads. And so that will be one potential thing, is that we probably, to do in monkeys, you'd have to either develop an entirely new protocol to differentiate stem cells into beta cells, or they have to look at approaches. So I think in the end, you really want to do a phase 1 trial. And we have a lot of things that have been done already, so they have blazed the trail, if you will. People have shown, like the islet transplant is FDA approved since summer 2023. Treg infusion has been shown to be safe since 2012, so about 15 years ago almost. And so the bits and pieces, the Treg infusion, the beta cell, the islet transplant are in place. Now, what's needed though is the approval for stem cell derived beta cell. And so companies like Vertex and now Sen as well are working on this. And so I guess the idea there is that I don't know how feasible it is in this specific approach where you co-engineer the stem cell and the Treg to these animal models that are not a humanized mouse or a humanized rat. There's no such thing, as far as I know, as a humanized monkey, right? Those are much larger animals. But that's a great question. I think something that definitely I think a lot about as time goes by. Like, how can we really prove this beyond any doubt that it works really well without going into patients? And I think it might be a short runway because I think we have to go into patients, basically. And again, we have the work from Sana that they are going into patients, right? And they're showing this, at this point, 1 month survival. So again, the trails have been blazed for us to do this kind of work as well.
Thank you. Our next question is, how did you choose truncated EGFRT as the unique ligand that Treg CAR recognizes?
Yeah, it's a great question. I think it took quite some thought. We're wondering, again, it has to be inert, so not really hurting the cells. Yes, we immunologically inert, meaning that it can't just be a bacterial protein, so it can't just, you could imagine engineering some weird, like, surface-bound GFP, but like that wouldn't work because then it would be a foreign protein. And then when I looked into that, and, being a primarily immunologist, we use a lot in a T-cell engineer, we use a lot of these truncated markers to sort T cells out. So in my lab, we use truncated C19, truncated EGFR, truncated NGFR, truncated C34. So we wonder, well, if these truncated markers have been kind of like stress tested for human stem cells, can we start maybe with one of these as our proof of concept ligand? And so that's really how EGFRT came about, was out from our work in using EGFRT and others as unique markers for human T-cell engineering. So we knew they were safe and they were inert. And then in the mouse model, there would be no issue of cross-reactivity, right? Because an anti-human EGFR CAR does not bind to mouse EGFR. And so, yeah, so that's how that came about, yeah.
Thank you. Are current safety switch satisfactory in case of secondary malignancy, especially considering allogeneic hypoimmune engineered cells?
Oh yeah, it's a great question. So this is why, having threaded on both worlds, I feel like. So I didn't mention, but as a PhD student, I worked on universal stem cells, and as a postdoctoral fellow, I worked on engineered Tregs. And so I've the engineering of both the graft side and the immune side. And I think the safety switch is really important on the graft side. So as I mentioned, the last thing you want is to have, beautifully protected graft. And then if there's a virus infection or there's cancer formation, you can't eliminate it. So there's a couple switches there. One is, this thymidine kinase based switches where you treat the patient with a drug and that would eliminate specifically the cells expressing that suicide gene, we call it. Another way is actually from this company, Sana, they mentioned CD47, and there is an anti-CD47 antibody. And so it seems as if you treat the patient, the recipient, anti-CD47, now part of that immune privilege is broken, and then the cells can be eliminated. From our side, I think it's actually intriguing, the possibility that our unique ligand can be used both to be recognized by protective CAR Tregs, but also by killer CAR T cells. So you can imagine a scenario where if you have, let's say, we have this new ligand A, and then we have this, a specific CAR Tregs that protect it, if that to be an issue with the graft, you could infuse, CAR T cells that would recognize and destroy that graft in a specific manner. And so we actually see the unique ligand not only as a way to be recognized by the protective CAR Tregs, but also as a built-in safety switch. So it will be working a bit like Sana C47 can be treated anti-C47, right, to remove those cells. You could also use these unique ligands as a target for destruction.
Our other question is, would allogenic human ex vivo 3D pancreatic beta cell analysis be helpful using CAR Ts from PBMCs?
That's a good question. I mean, I think that there's definitely a lot of excitement in the organoid field, and for the biology, I think it's fascinating. You can even you can do other organoids, 3D cultures. You can do pancreatic slices as well. They preserve quite some physiology if the slice is thick enough to look into metabolism and gene expression and all this. For us, though, I think, as I said early on, I think what maybe fascinates me about the immune system is that it's really everywhere in your body, right? It's not just in one place. And so at the in vitro can have a hard time capturing that in vivo does is a trafficking and survival, right? We inject these cells into mice intravenously, and they have to find their way to the graft, and they have to survive on their way there, and they have to survive while they're there. Whereas with 3D models in vitro, they will not recapitulate that. That said, you can definitely do quite some interesting work. For example, I mentioned that part of our work now is involved in the design of the CAR receptor itself. And so I think for this, we're starting all in vitro, right? Before we go into the in vivo functional studies, we can all in vitro, we can see how tight it binds, what cytokines are being made, how does it affect the beta cell physiology and phenotype and morphology and function. And so that's where I see that. And then, of course, I think 3D was mentioned because the 3D culture will be more representative of what happens in humans as just having a monolayer. But yeah, so it's somewhere in between. But again, my view still is that to look into efficacy and safety, you still need the animal studies. And so actually in my lab, we've developed some CAR T cells as an example that in vitro, they worked pretty great. They killed the target tumor cells and all this. And then when I put them into mice, actually they're toxic to the mice. And so I think to look into safety and efficacy, you need to have a full living organism to get a better picture of what's going on.
Fantastic. I think this might be our last question. Just wondering, how effective do you think this method will be for different types of organs?
Oh, that's a great question. Yeah, so we started with this one, and my background is on type 1 diabetes, and so is Dr. Russ's background as well. And so we started there. Also, the field has, over the past almost 20 years now, has built the tools to make stem cell-derived beta cells. And so there was a natural first place and indication to look at. But that said, we also have a collaboration with a cardiac organoid lab, for example. And heart disease being the #1 killer, it's a very important application to go after. And so that's a great question. So we are exploring the idea that, what is any specific biology we're not aware of, or can this be a universally applicable approach for any stem cell-derived tissue transplant? And so we'll find out. So again, the beta cells, really, it's about the pancreas, and or if it's a liver, the portal vein transplant. So again, location could play a role, right? Different sites in your body have different inflammatory, anti-inflammatory potential. So the liver tends to be very tolerogenic, right? So actually in the cancer world, if your cancer metastasizes to liver, it's very bad news. And so we wonder, for example, now in the heart. So if the challenge in type 1 diabetes is hyperglycemia, and also I didn't mention, but Tregs also don't love insulin. If you knock out the insulin receptor in Tregs, they seem to be happier than if they have it. In the heart would be ischemic perfusion, so lack of oxygen, severe hypoxia and anoxia, and also just fibrosis, right? Every time you injure the heart, those beating cardiomyocytes are replaced with non-beating fibroblasts. And so, yeah, it's a great question. We're very interested in this, in seeing how applicable it is to multiple disease contexts.
Great. Well, thank you so much, Leo, for that really, really fascinating talk. That was great. And thank you, everybody, for joining us. And thanks for the questions, and thanks for the discussion. I hope that, we can follow this research, keep up with your publications, and see what updates we can see in the upcoming months and years. Thank you so much.
Thank you. Thanks for the opportunity.